Today we're going to be talking about fraternal twins and identical twins. Let's go ahead and start with the easier of the two, which is going to be your fraternal twins, also known as dizygotic twins. Fraternal twins are going to be the result of fertilizing two separate eggs. Thus the word di in zygote refers to a fertilized egg, a single cell. So what's going to happen here is this. Normally a human female has two ovaries, and 1 month one ovary releases an egg, and the next month the other ovary releases an egg, and we go back and forth. But once in a while what happens is both ovaries release an egg. And now we've got two eggs. And upon introduction of sperm, both eggs are going to get fertilized. And this is going to result in two completely genetically different babies being born. Now with identical twins, also referred to as monozygotic twins, mono meaning one, you're going to get two babies as a result of a single fertilization event. Alright, so you're going to end up with one embryo, and that embryo is going to split and form two babies. Let's go ahead and talk about that process, because it's a little bit different depending on what type of animal you are. In one group of animals called protostomes, identical twins can't occur through the splitting of the embryo. However, in deuterostomes it can. So let's go ahead and draw this. I'm going to go ahead and I'm going to start with a morula. And our morula is going to be at the level of 4 cells. So this is our morula, our embryo. And remember, an embryo is going to be 2 or more cells. So we've got a 4-celled embryo here. In protostomes, what's going to happen is this. This embryo is going to split. Sometimes this happens. The embryo splits for whatever reason. And that's going to result in 2 separate embryos. However, in protostomes, the cells become genetically determined very early. And in fact, this is going to be called determinate cleavage. Because this form of cleavage results in cells that become genetically determined or genetically set very early in embryological development. So what I'm saying here is once this embryo splits, these 2 cells, once they divide, can't reform these 2 cells that they're missing. And these 2 cells can't reform these 2 cells that they're missing. So if these 2 embryos are allowed to further develop, both embryos eventually end up dying because they're missing their other half. In deuterostomes, things are going to be a little bit different. So let's go ahead and write deuterostomes down here. And go ahead and draw our embryos again. Our embryos have split. And in this case, they've split into 2 embryos, but we could get 3 embryos and sometimes even 4 or more embryos. So now we have 2 embryos. And the process here is going to be referred to as indeterminate cleavage. So let me go ahead and write that down. In this type of cleavage, the cells don't become genetically programmed quite as early. They will eventually, but not quite as early as in protostomes. So when the embryo splits, these 2 cells, this embryo, can, when it undergoes cell replication, can reform the 2 missing cells. And this embryo can also do the same thing. When these cells replicate, it can reform these 2 missing cells. And if these 2 embryos are allowed to further develop, they go ahead and they completely develop, they're completely normal, and they're going to be genetically identical. And this results in identical twins. Now, I just want to make sure that you don't confuse determinate cleavage with determinate growth. Determinate growth is when an organism at some genetically pre-programmed time stops growing. Usually in humans, we stop growing anywhere between 16 and 18 years old. And then we just don't grow any taller anymore. With other organisms, we have indeterminate growth. And indeterminate growth means that the organism continues to grow throughout its entire lifetime. Usually as the organism gets older and older, the growth slows down, but still the organism continues to grow throughout its lifetime. Plants are a good example of indeterminate growth because they continue to grow throughout their entire lifetime. Not the leaves, but the stem and the roots continue to grow throughout their entire lifetime, albeit slower and slower as they get older. Again, don't confuse determinate growth with determinate cleavage, and don't confuse indeterminate growth with indeterminate cleavage. They mean two different things. And that's going to be it for fraternal twins and identical twins.